All right, welcome back to our discussion of meiosis, chapter 13 of Campbell's biology. Today we're going to be moving into concept 13.3. Meiosis reduces the number of chromosome sets from diploid to haploid, and we will go into glorious detail about how that happens. All right? So, like mitosis, meiosis is preceded by the replication of the chromosomes. Okay? When we show you diagrams, oftentimes we do not point this out, but always remember that just like meiosis is preceded by interphase, where we have G1, S phase, and G2, meiosis is also preceded by that. So the very first step is to replicate our DNA. Meiosis takes place in two consecutive cell divisions. So we go through meiosis 1 followed by meiosis 2, right? So in mitosis, we have 46 chromosomes, so we are a 2N. But when we replicate our DNA, we are now at 4N, right? The same thing is going to happen here. And when we go through meiosis 1, we will be back down to 2N, and then further reduce it to 1N when we go through meiosis 2, because there is not a second round of DNA replication happening between meiosis 1 and 2. Now, the two cell divisions will result in 4 daughter cells rather than 2, which occurs during mitosis. And again, each daughter cell will only have half as many chromosomes as the parent. So here we will have 1N cells. So just as a schematic, here we have a cell that has 2 chromosomes. These are both chromosome 1, this one from dad, this one from mom. The very first step is to go through interphase, where we replicate that DNA. That produces these sister chromatids. So now we have 2 copies of dad's chromosome 1 and 2 copies of mom's chromosome 1. In meiosis, we will go through a round of cell division, which will separate that, producing a cell that now has only 2N, but then we will go through a second division that will reduce it to only 1N. So here we have 1N, or 1 copy of chromosome 1 from dad, 1 copy of chromosome 1 from mom. In this cell, we only have 1 cell with 1 copy of chromosome 1 from dad, 1 copy of chromosome 1 from dad. Here we have a cell that has 1 copy of chromosome 1 from mom and 1 copy of chromosome 1 from mom. Right? So now we have 4 cells and they have half the amount of DNA. So again, here we have 2N. When we go through interphase, we will always end up, we're going to go down to 1 end. So here we will end up with 4N until we go through that first division. That will bring us back down to 2N, and then finally down to 1N. So let's talk about the steps here. The steps carry out the same progression. So you can see that there's going to be a prophase. You can see that there's going to be a metaphase with a metaphase plate, and then there's going to be an anaphase where they're pulling apart. And then we have telophase and cytokinesis happening, right? So let's go into the steps here. Now, in prophase 1 of meiosis 1, so remember there's a meiosis 1 and 2. This is just 1. So prophase 1 of meiosis 1, something special happens. We have chromosome 1 from mom and chromosome 1 from dad. They are going to seek each other out and form together this 4 chromosome unit called chiasmata. Okay? So we're going to have both sister chromatids of 1 from mom, both sister chromatids of chromosome 1 from dad, and they're going to link together. Same thing is going to happen for chromosomes 2 and chromosomes 3. Same other all steps are happening here. So we have the nuclear envelope breaking down, and we have the spindle forming. Now, something unique happens when we create this cluster of all the chromosomes 1 from both mom and dad. Okay? So this process is what we call crossing over. So here we have both of those chromosomes from the paternal, and here's the maternal sister chromatids. So we have 1, 2, 3, 4 copies of chromosome 1, 2 being from dad, 2 being from mom. We will then intentionally break the DNA at various points along one of the strands of chromosomes. Okay? When we do that, it is not a flush break. So remember that each strand of DNA has a reading strand and a complementary strand. When we break, there will be an overhang. That overhang will then reach over and find the complementary strand of, in this case, the maternal will seek out the paternal overhang. And the same thing is going to happen over here. At the same time, we are going to be forming a very tight junction between these chromosomes. The reason why we form this tight junction is we are breaking the DNA. We do not want to lose pieces or put them back together wrong, so we need it very tightly adhered. When we glue it back together, you'll notice that this strand of DNA from our dad is continuous and is identical to the DNA that came from our father. This strand is continuous with the DNA that came with mom. This strand, however, is DNA from mom, then all of a sudden DNA from dad, and then all of a sudden back to DNA from mom. This one is the counterpart to that, so now we have a little bit of DNA from dad, then over to mom, then back to dad, right? So we're creating this hybrid form of these chromosomes. So we will have one intact chromosome from dad, one intact chromosome from mom, and then we will have 2 chromosomes that are each complementary hybrids of both, right? So maybe it's better to look at it this way. Let's blow up on this. So here we have metaphase, where we're going to have this so this occurs in prophase, then we go to metaphase, and we're going to break these apart. And all of a sudden here you can see chromosome 1 that is a full copy from mom, and chromosome 1 that is mostly mom, but a little bit dad. Over here we have chromosome 1 from dad, full copy, and a second chromosome 1 that is mostly dad with a little mom. And you can see this happens all along the way. When these finally get separated in meiosis II, we'll now have a unique combination where no 2 of these cells are completely identical. So here we have a chromosome 1 from mom intact. Here we have chromosome 1 that is mostly mom but a little bit dad. Here is all dad and mostly dad with a little bit of mom. And you can see that creates variety in all of these cells. So let's go back to where we were talking about here. We have prophase 1. We have crossing over occurring in prophase 1. That is the most important part. Here, when we go into metaphase, we will form a metaphase plate, but instead of having individual chromosomes line up here, we will have all of the chromosomes together in this chiasmata coming along this plate. So instead of an individual chromosome, we have 4 chromosomes lining up here. And what we do is we will separate the sister chromatids from the other sister chromatids. But remember, one of those chromatids are now a hybrid. Okay? So that gets us through the metaphase plate. We then, of course, go through anaphase. We then pull those apart. And then we go through telophase and cytokinesis. Now, instead of going through another round of interphase, we will just progress immediately to another round of division. Okay, and here sometimes you see a nuclear envelope reforming, sometimes you don't. It depends on the scenario. So going into meiosis 2, we have prophase 2, and metaphase 2, and then anaphase 2, and telophase and cytokinesis 2. Okay, so here we do get individual chromosomes, chromosome pairs lining up along the metaphase plate. Those get pulled into single chromosomes. And now we have haploid cells occurring in what is now 4 daughter cells. So looking at the whole schematic here, again, we have meiosis 1, we have prophase. This is where we get these tetramer forming, so these all 4 chromosomes coming together, forming this chiasmata. We get crossing over occurring, producing these hybrids. We then go into metaphase, and here we have all 4 chromosomes as opposed to individual chromosome sister pairs in there. We pull those apart. We then go into telophase and cytokinesis, and then without going into another interphase, so no additional DNA replication, we immediately go into prophase 2, metaphase 2, anaphase 2, and then ultimately forming all 4 haploid daughter cells. So stopping to compare mitosis and meiosis, which is something that is helpful for us to do. Meiosis conserves, or mitosis conserves the number of chromosome sets, producing cells that are genetically identical to the parent cell. So you get one cell dividing or replicating its DNA and then dividing cleanly in 2, giving 2 daughter cells that are genetically identical. Meiosis reduces the chromosome number from 2 to 1 and produces 4 genetically distinctive daughter cells. So here, looking at side by side, here we have a parent cell. In this case, it's going to go through mitosis. So we have 2N. Here we're going to go through a phase where we are 4N. So we duplicate all the DNA. We are now 4N. Here we line up with all of the individual chromosome sister pairs along the metaphase plate. Over here in meiosis, we replicate the DNA the same, but now we get these chiasmas coming together, and we get crossing over occurring. Now we go through metaphase 1, and we have these tetramers pulled together here. Over here, we will then split that DNA neatly into two 2N cells. Here we will split and have, two 2N cells, but then we will immediately split them again, producing 4 1N cells. And notice that while these are genetically identical to the parent cell, both of them, each one of these is not only has half the amount of DNA, but notice that they are genetically distinct. This has chromosome 1 from mom. It has chromosome 2 that is mostly mom with a little dad, and a chromosome 3 that is mostly dad with a little mom. This one, chromosome 1 is mostly mom with a little dad, chromosome 2 is all mom, and chromosome 3 is all dad. And you can see all the different combinations we can get here. So, things to keep in mind is things occurring in meiosis 1, synapsis and crossing over occurs in prophase, and you have homologous pair at the metaphase plate occurring. So, again, keep in mind the various steps and how these things differ. Be able to compare and contrast mitosis and meiosis. What's different? What's the same?